Good morning. We have a short lecture to end the week. I'm going to introduce the anatomy part of the urinary system, and then we'll wait until next week, until most of you have taken the current exam, to get into the physiology. I don't want to start throwing a lot more numbers and gradients and calculations to you until you have taken that exam. So we will go over the, start letting a few more people in. We'll go over the anatomy, and this is also a good review for you. Have a lab exam next week, so the material that we'll cover today should be helpful in that as well. All right. So the kidneys are part of the urinary system, and that's where we're going to start with our a lot of our anatomy and our physiology. Is the functional unit of, we'll talk about the nephron, which is the functional unit of the kidney. And then once urine is produced, it's transported in the ureters, and it is stored in the urinary bladder and then transported out of the body using the urethra. And we're going to be going over all of these structures in much greater detail, as you would imagine. And the kidneys have several important functions. The main one, just every minute of every day, is to regulate water volume and total solute concentration. A lot of those concepts pretty much go hand in hand, especially when you're talking about sodium. Where sodium goes, water follows. This will also assist in regulating ion concentrations in the extracellular fluid. We do this, so it's not only sodium that's balanced, but also things like potassium and magnesium and calcium and urea. There will also be the kidneys' role in acid-base balance, which we will get to in about 2 weeks, and that will be mainly in affecting the bicarbonate concentration. The kidney tubules, especially in the distal convoluted tubule, will be a place to excrete metabolic waste, toxins, and drugs. And as we've already discussed in blood, the kidney produces erythropoietin to help regulate blood pressure, to get more red blood cells to be produced, and to stimulate the release of reticulocytes into being released to form erythrocytes. And then for mean arterial pressure, when it decreases, renin production and secretion is going to occur. And I just realized on the slide, these are reversed. Producing erythropoietin regulates red blood cell production, and renin regulates blood pressure. So I'll correct that and put up a better version. Activating vitamin D, carrying out gluconeogenesis. We probably won't be going into these two as much. So we'll be focusing mainly on the top 4 because we've already talked about number 5, and we may or may not mention numbers 6 and 7. Okay. Where are your kidneys? Your kidneys are retroperitoneal. So they are going to be behind the peritoneal cavity, which is what contains your organs like liver, pancreas, stomach, intestines. So if you take those out, that's what this peritoneal cavity is. And then the kidneys sit behind it on either side of the vertebral column. The right kidney is slightly lower, and so it is more vulnerable to injury. There are two adrenal, also known as suprarenal glands, that sit atop each kidney. They are not related in function to each other. It's just a structural relationship that occurs during development. The adrenal gland tissue just kind of relocates and squashes on top of the kidney. It's just a good place for it to be. If you've ever heard of like a kidney bean, that's the shape of a kidney. It has this concave medial surface where we get the hilum, which leads to the internal space of the sinus. We have ureters, renal blood vessels, lymphatics, and nerves entering at that concave section of the kidney. There are 3 layers of supportive tissue that surround the kidney. The one that is right around the kidney is called the renal capsule. So it's like that when you hear of a kidney bean, it's got that dark kind of brown color. That is the renal capsule that surrounds the cortical and medullary tissue. And then the kidney sits in this perirenal fat capsule. So it's encapsulated in fat to protect it. And then there is a fibrous capsule that is going to anchor it into its cavity. So 3 layers of protective tissue. So the capsule, the perirenal fat capsule, and then that renal fascia. So now we're going to talk about macroanatomy, so large sections of the kidney. The kidney is divided into 2 main layers, even though it has this 3 distinctive regions. The 2 layers of the kidney are the renal cortex and the renal medulla. For the cortex, it's this outer band of tissue, and it's going to contain things like the glomerulus, the proximal convoluted tubule, the distal convoluted tubule, and then it also goes down in between. These are called medullary pyramids. The cortical tissue comes down and forms renal columns. This inner layer is called the renal medulla, and it is organized in pyramids. So this entire layer is called the medulla. Each segment is called a pyramid. Pyramids are the loop of Henle. The loop part is actually in the medulla, and then the majority of the collecting duct, so most of it runs through the medulla. So the medulla, when you look at it under a histological slide, these are collecting ducts that are running down. The medullary pyramid ends in a little structure called the papilla. So it's this end. And then the papilla is going to drain into a minor calyx. A couple of minor calyces get together and they will drain into a major calyx. And then there are a couple major calyces and they drain into the renal pelvis, and the renal pelvis drains into the ureter. I think that covers most of the cortex and the medulla. So we have our cortex is a superficial region. It does come down to form columns and separate those renal pyramids. The medulla is a layer, and each it's defined as pyramids. Pyramids are made up mainly of collecting ducts and then also quite a few blood vessels, the vasa recta. It ends in the papilla, which then drains into a minor calyx, a major calyx, the renal pelvis, and the ureter. The renal pelvis is the funnel-shaped tube, continues with the ureter, and it is going to collect, filtrate from, actually I guess it's urine at that time, from the major calyces and funnel that into the ureter. So you do need to know the flow of urine and be able to identify those structures. Your kidneys receive 1/4 of cardiac output at rest. So each kidney is going to receive about 600 mils of blood. So together it's 1.2 liters per minute. So that is a tremendous amount of blood flow. And that's because the kidneys will need access to it to produce filtrate and adjust its composition, as well as to monitor the ion concentration in the blood. The arterial flow, we're going to start at the renal artery, branch into segmental, into interlobar, arcuate, and then cortical radiate, which are sometimes called interlobular. So the blood flows in through this renal artery. Then the renal artery will branch into 5 segmental arteries. The segmental arteries further branch into interlobar arteries. And then this arcuate artery is going to be very important. The arcuate artery defines the cortex from the medulla. So superior, or I guess it would be more superficial from the arcuate artery, it's going to be the cortex, and deeper will be the medullary layer. So these arcuate arteries are going to be, I always think of them as being horizontal, because then the cortical radiate arteries come off of it like spokes. These cortical radiate arteries are also important. So we've got our blood flow coming in from the renal artery, divides into segments and in between the lobes. Then we get this arcuate artery that defines the cortex from the medulla, and then cortical radiate arteries come up. Well, off of these cortical radiate arteries are the afferent arterioles that start this whole process of filtration. We'll talk about, there's another picture that gives you a much better view. And then if you've had lab, I think most of you have, you've been able to see the models and look at how, and look at these up close in model form. The afferent arteriole is going to lead to the glomerular capillaries. The afferent arteriole is larger in size than the efferent arteriole. And this difference in size is going to cause an increase in pressure at the glomerular capillaries, and that pushes the filtrate out through those fenestrated capillaries. So it's like big supply coming in, lower supply going out, increases the pressure, and that's going to force out the filtrate. The efferent arteriole then is going to join up to the peritubular capillaries or the vasa recta, and then those are going to feed into cortical radiate veins if they're in the peritubular. We're talking about peritubular capillaries, which then feeds into the arcuate, the interlobar, the renal vein, and inferior vena cava. We'll also, I don't know if you, well, hopefully you remember from veins, they're not as defined, like their pathways aren't as distinct. So that's why you're getting a few arrows. Some of these are going to drain into the cortical radiate. These are the peritubular. The vasa recta are going to just drain into the arcuate. And then there are no segmental veins. So the venous structure is a little bit more fluid, but it is good to remember that the renal vein flows into the inferior vena cava. Renal plexus, there is a substantial number of autonomic nerve fibers that are especially sympathetic ones that are going to regulate blood flow, and that regulates filtration rate as well. We'll talk about that much more in detail later. Nephrons. So nephrons are the structural and functional unit of the kidney. There are about 1 million of them per kidney, and there are two main parts. The renal corpuscle is going to be made of the glomerulus and the glomerular capsule. So the glomerulus is this tuft of fenestrated capillaries, and it's going to produce filtrate. So we have our arcuate artery, and it's going to separate the cortex from the medulla. Then coming up from the arcuate artery, we have a cortical radiate artery. Coming off of that cortical radiate artery, you're going to have several afferent arterioles. And this would be much closer. I'm just giving myself space. So the afferent arteriole is large, and it is going to then join up with the glomerular capillaries, sometimes just referred to as the glomerulus. Then we have a smaller efferent arteriole that ends. Because of the difference in size, as well as that these are both arterial systems, so they already have high resistance, that is going to push filtrate, which is essentially plasma minus the proteins, should be. Sometimes if there's protein in the urine, then that indicates a problem, often at the level of either blood pressure or glomerular capillaries. And then that filtrate is going to get collected into Bowman's capsule, also known as the glomerular capsule. So as the filtrate is produced, it is caught in this glomerular space and funneled into the proximal convoluted tubule. So the renal corpuscle is the glomerulus, which are these capillaries that are fed into by the afferent arteriole, then it exits out of the efferent arteriole. The filtrate is captured by this glomerular capsule. The glomerular capsule has two layers. So it has a kind of a more structural layer, this part, okay, before it goes into the proximal convoluted tubule. This is the parietal layer here, and it's made up of simple squamous epithelium. Then there is a visceral layer, which if you look at the model from lab, it's like the glomerular capillaries are, some of them are red and some of them are white, and that white is this visceral layer of the glomerular capsule. These are called, the majority of the cells are podocytes, which have little feet on them, and the feet will open and close to help regulate and the filtration rate. Once filtrate is produced, then it's going to enter the tubule system. The beginning of the tubular system is that proximal convoluted tubule. So we have proximal and we have a distal, and the proximal means that it is closest to the renal corpuscle. Distal means it's furthest away, but it will also be the one that leads into the collecting duct. So we usually start, afferent arterial, glomerular capsules producing filtrate, captured by the capsule, kind of like a baseball glove catching a baseball. Well, baseballs, lots of them producing lots of filtrate. And that filtrate is going to be funneled into the proximal convoluted tubule. The proximal convoluted tubule is going to reabsorb 98 to 99% of what enters it. So it is major function is reabsorption. And we will go into the physiology in much, much greater detail next week. But the proximal convoluted tubule is made up of cuboidal cells with dense microvilli. So these dense microvilli form a brush border, and that brush border is very, very thick. It is so thick that the lumen gets this like fuzzy looking appearance, because it's almost like you filled it with like a cotton ball to absorb everything that's going through it. It does play a minor role in secretion, but absorption is really its function. If we didn't have a proximal convoluted tubule, we would be unable to maintain the fluid in our body. So proximal convoluted tubule, really, really important. They do also have large mitochondria, and that's because some of this reabsorption is through active transport. So we do need ATP being produced to transport some of the substances back. Okay, from that proximal convoluted tubule, then we go into the nephron loop. Okay. So we have, this is the arcuate artery, cortical radiating artery, afferent arterial leading to the glomerulus, efferent arterial, which most of these will then lead back to the peritubular capillaries. The glomerular capsule has the parietal layer here on this outside, kind of the catcher's mitt, as well as a visceral layer, which attaches to the glomerular capillaries to help with filtration rate. Once the filtrate is made, pushed out of those capillaries, it enters the proximal convoluted tubule. So on one side, you have an afferent and efferent arterial, and on the other side, you have filtrate entering into this proximal convoluted tubule. The proximal convoluted tubule leads to the loop of Henle. The loop of Henle has a descending limb, a loop section, and then an ascending limb. And next week, we'll talk about these thick and thin segments more, but why some are thick and why some are thin is going to depend on what tissue type they're made from. Some are more cuboidal, some are more columnar. We even have some that are squamous. So smaller cells lead to a smaller limb, and we'll talk about their function next week. For now, just focus on the anatomy. And then in lab, if you make sure, because each model is set up a little differently on the nephron board, make sure that you're tracing the pathway from that glomerulus so that you know exactly which is the proximal convoluted tubule, which one is the distal, and if you're on a descending limb or an ascending limb when you have to identify structures. Just a little hint. Okay, the filtrate goes down the loop of Henle, back up the loop of Henle to the distal convoluted tubule. The distal convoluted tubule is going to be a major site for secretion. So there are certain substances that we don't want making this large journey through our delicate tubule system. So we will secrete more nasty things into the distal convoluted tubule, which is then going to dump into the collecting duct. The collecting duct, so there are 1 million nephrons and 1,000 collecting ducts. So roughly, right? So the collecting duct is going to receive filtrate from many, 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 many nephrons. The collecting duct is made up of 2 different cell types principal cells and intercalated cells. We'll talk about those more next week and their role in acid-base balance and maintaining water balance. Okay, I think that's most of that. Okay, so proximal convoluted tubule leading to the loop of Henle has a descending and then an ascending. The descending thin limb is made up of simple squamous. That's what makes it thin. And then the thick ascending limb is cuboidal or columnar cells, so bigger cells. This slide is one that you also should be observing in lab as well. It's set up underneath the microscope. These are the glomerulus, so the glomerular capillaries. The white part is the glomerular space. And then this layer here is the parietal layer of the glomerular capsule. So the glomerular capsule, the parietal layer is made up of this simple squamous, and it's this one. There on this slide, it also shows like a distal convoluted tubule. You can see how it has this nice clear lumen versus this is like a proximal convoluted tubule, this one and this one and this one. And you can see how it's so fuzzy. It's because it has such thick microvilli to absorb all of the ions and amino acid and glucose and sometimes calcium, everything that's coming through there, magnesium. So the distal convoluted tubules have this clear lumen. Proximal convoluted tubules have a fuzzy lumen. As I mentioned, the collecting ducts, they receive filtrate from many nephrons. Then they are going to run through the medullary pyramids and end at the papilla. Then once at the papilla, it delivers urine into the minor calyx, to the major calyx, to the renal pelvis, to the ureter. We'll go into the two cell types much more in depth next week, but collecting ducts do have two main cell types. Principal cells, which are going to be involved in water and sodium balance. Like I said, those two tend to go together. And then the other population are intercalated cells, and we'll talk about how they assist in acid-base balance. There are two main groups of nephrons. 85% of nephrons are cortical nephrons. So when we are looking at the different, so we have our cortex up here and our medulla up here. For our cortical nephrons, the glomerulus tends to be up higher, and then you get the loop, and it just loops a little bit into the medulla and then to the collecting duct. Okay. For juxtamedullary nephrons, the glomeruli tend to be closer to that border, and then the loops go way down into the medulla until then you're going to come over. Okay. Cortical nephrons are your typical nephrons doing the bulk of, kidney functions. Juxtamedullary nephrons are going to be the ones that are involved in producing a concentrated urine when we need to save water volume, increase extracellular fluid volume. So cortical nephrons, 85%, they are going to live primarily in the cortex with a little bit of a loop down to the medulla. Juxtamedullary glomerulus tends to be much closer to the border. The convoluted tubules still live in the cortex, but the loop goes way down into the medulla. This diagram also illustrates how there are two capillary systems involved with the kidney tubules. If you remember, we have a lot of reabsorption and some secretion going on. And if the kidney tubule cells are pulling items out, they need to have somewhere to go. And for the convoluted tubules, they have this tight, close relationship with peritubular capillaries. Whereas in the medulla, when we're talking about these long loops of Henle, the capillaries are the vasa recta. The two groups of capillaries that are going to assist the kidneys as they do their jobs of mainly of reabsorption, sometimes of secretion. For capillary beds, we've talked about these, but I'll review them again. The glomerulus is going to produce filtrate. It has that afferent arteriole going in, glomerular capillaries. This makes up the glomerulus, and then an efferent arteriole going out. We get filtrate due to the difference in pressures. Peritubular capillaries are up here in the cortex. Vasa recta are down here with the loop of Henle. The glomerulus, these are specialized for filtration. They are fed by an afferent arteriole. Blood exits via an efferent arteriole. The afferent arteriole arises from those cortical radiate arteries, and then the efferent feed either into the peritubular capillaries in the cortex or the vasa recta in the medulla. Blood pressure in the glomerulus is high because the afferent arteriole is larger in diameter, and arterioles in general are high resistance vessels. Peritubulars, capillaries are low pressure. These are kind of your typical capillaries. They are porous because they are going to be receiving a large amount of water and solutes. The vasa recta, these are long vessels. They are going to parallel the nephron loops of these juxtamedullary nephrons, and they will assist in the formation of concentrated urine. So they'll also be reabsorbing water, just in a different location. Each nephron also has a juxtaglomerular complex. If you looked at the model, we had our afferent arteriole, and then we had little, actually kind of branching efferent arterioles. These, the afferent arteriole, you'll see, has these specialized cells on the lab model. These specialized cells are part of that juxtaglomerular complex. The juxtaglomerular complex is going to be made up of 3 different cell types. We have one that's part of the afferent arteriole, one that is part of the ascending limb where it becomes the distal convoluted tubule. I know, because that's not complicated. So you've got the ascending limb coming up, and it's about to turn into the distal convoluted tubule. And there are columnar cells in here. And then there is glue that essentially holds them together, called mesangial cells. So there are 3 cell types in this juxtaglomerular complex. They are going to be used to monitor salt concentration, as well as help in the regulation of blood pressure. So, whoops, just to tell you another slide, right? The 3 cell populations are for, I'm going to start, I always like starting with the afferent arteriole. So we have granular cells of the afferent arteriole. These are enlarged, smooth muscle cells. This is, on our models, it's always shown as the afferent arteriole. In real life, sometimes it involves the efferent, but just to keep things straight, we always are going to just say that they're on the afferent arteriole. These are going to act as mechanoreceptors to sense blood pressure. And if you remember the renin-angiotensin-aldosterone system, if you're not remembering that, I would recommend that you go back and review it. But we get a decrease in blood pressure, an increase in renin release, and then that creates, through that cascade, angiotensin 2, which then has its own 4 effects. So granular cells are located at that afferent arteriole for where the ascending limb turns into the distal convoluted tubule. So you'll also see on models, they're kind of, sometimes it'll label that part of the model distal convoluted DCT, but they are where this ascending limb turns into the distal convoluted tubule. They are going to, so it's called the macula densa. They are made up of columnar cells that contain chemoreceptors that are going to sense sodium content. And then to keep this all nice together, we have mesangial cells that do have gap junctions to help connect physically and ionically the macula densa and the granular cells. So that is the trio for the juxtaglomerular complex, sometimes called the juxtaglomerular apparatus. Again, you got to love anatomy, and we have to name things at least twice because once just doesn't seem to be enough. The 3 populations, these cells, the macula densa cells, they're coming up from the ascending loop, they're about to turn into the descending, the distal convoluted tubule. These macula densa cells are chemoreceptors to monitor the sodium content that's coming from the ascending limb. And then they are connected through these mesangial cells to the granular cells of the afferent arteriole, which are going to act as mechanoreceptors to regulate, monitor blood pressure. So this juxtaglomerular complex or apparatus consists of these 3 cell populations. And this is where we end for today, and we pick up next week